everybody to 8th grade and to Integrated Science 2. For those I have not yet met, my name is Dr. Ackerson. I am one of the two 8th grade science teachers along with Mr. Kell. And this is the first of a couple of different videos we're going to ask you to watch throughout the year. It is a quick review of the high points from Integrated Science 1. Now this is not to say that everything in Integrated Science 1 wasn't important, but I'm going to go over the things that are going to be important for us in Integrated Science 2. So if you're watching this video, obviously it is for review. There are 7 homework problems for Dr. Ackerson. Mr. Kelp has a slightly different homework assignment. Feel free to try those before you watch this video, and then fill in your knowledge with the video if you can't answer the questions. All right, let's go ahead and get started. All right, so basically what we're going to go over is microscope use, the idea of molecules, elements, and reactions. We're going to talk about cells and the difference in plant and animal cells and the requirements for them. Okay, so this should look pretty familiar. This is a compound microscope, and I've put this picture here because it has all of the relevant parts. Now, nowhere in this year will you be graded on the parts of the microscope. That you've already done that. That's already done. However, I would familiarize yourself with those parts again if you have forgotten them, because as we do microscope work in this year, I will refer to these pieces as if you know what you're talking about. So in other words, I will refer to things like the eyepiece. Objectives is very important. I'll talk about the nose piece, the arm, the base, light source, coarse adjustment knob, fine adjustment knob, stage, and stage clips. Okay. Incredibly important when using a microscope is to really understand what a microscope is and how it works. So if you allow me just a moment, I have a pen here. Okay, so a light microscope starts down here, right? And, cleverly enough, directs light up that way. The light then comes through the diaphragm here, condenses into a fine beam, and comes up through here. So that little opening in the stage. This is where the slide would be. Okay? Now, from there, if an objective is pointing straight down, it's going to go through the objective. Now, something that we're going to learn in second semester in physics is that light doesn't bend. Light always moves in one direction without bending unless it hits something that will bend it. So, in other words, this light will go on and on and on and on in this direction unless it hits something that's going to bend it. Anybody know what that is? And hopefully you said mirror, because that's right. Somewhere in here, there is a mirror. Finely polished, highly specialized, very sensitive mirror that is going to bend light, how like that, and up like that. So how else will you be able to see over here? Not your eye should be over here, looking down. That's my eye. Okay, so because there is a finely, precisely positioned mirror up here in this area here, you have to be extremely careful with microscopes, because if you jostle them, if you hit them too hard, if you knock them over, bang them on a table, it is possible that you could misalign this mirror in there, and then what you have is essentially a $2,000 paperweight. Because there is no way to reposition that mirror. Well, I guess there is, but it would be more expensive to actually hire somebody to come out and reposition that mirror than it would be to buy a new microscope. So we want to be incredibly careful with our microscopes, only grabbing them by the arm and the base. That's 2 hands now, and never putting them on a table closer to the edge than 1 hand, than 1 palm width. Also another thing, okay, so that you will find a piece of glass here and a piece of glass here. Both of those are what magnify the image. If you touch this part right here with your fingers, you're going to get oil in there, because your fingers naturally produce oil. I don't care how clean you are. Your fingers will produce oil to protect them against bacteria, because your oil contains what's called RNase, which will destroy the RNA. We're going to talk about what that means too, in bacteria, fungus and parasites, keeping you somewhat safe. Your skin is actually one of your best defense mechanisms. The point being, if you put the oil on the bottom of that objective, one of two things is going to happen. One, first, you're going to cloud the lens, and I'm going to have to come through with lens paper and clean it. It's a pain in the butt. I don't want to do it. Please don't make it. Second, if any of that oil gets stuck in the little crevices in there, you can get fungal or algal growth. If either one of those things happen, again, $2,000 paperweight. So you want to be very, 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 very careful never to touch the bottoms here with our fingers. If we do want to turn from one objective to another, we always want to use the revolving nose piece to do that. Last thing I'm going to talk about on this picture are the coarse adjustment knob and the fine adjustment knob. The coarse adjustment knob is when you're on 4x only, the objective 4x, also known as the scanning objective. Right? This knob is for every other objective, right? Because this knob moves the stage a lot less. And the reason for that is just sheer distance. When you have a slide down here, and you're on one of these really long objectives, if you move the stage too fast, you will crack the slide and maybe even the objective, and that's bad. Another note on microscope use. Let's say you see something like this, right, in your microscope, okay, at 4x. If you want to zoom in, you've got to make sure that whatever you want to zoom in is in the center of your focus. Because what's going to happen is as you go from one objective to another, the field of view gets smaller and smaller like a bullseye. So now, all you'll see is this area, but at a higher magnification, which means you'll lose everything on the outside. So make sure when you are moving from one objective to another, what you want to look at is in the dead center of your field of view. Okay, so just a basic review. Things that you should do with a microscope. Again, always start on the 4x or scanning objective. Always carry a microscope with two hands using the arm and the base. Use the coarse focus knob for the lowest objective only. Then use the fine focus knob. And remember to take all slides off, unplug, and wrap up your cord and cover whenever done. Do not ever touch the glass on the eyepiece or the objective. We just talked about why. Don't ever carry a microscope in one hand. Do not shake or drop a microscope because that mirror. Never leave a microscope on. You could potentially destroy my slide, burn out my light bulb, start a fire, or all three. And never, ever, ever use a microscope when you have dirty gloves on. Because again, you can get some of that dirty stuff inside the objectives, and that's just gross. All right, a couple of other things for microscopes. Words that I am going to use, that I'm going to expect that you know how to use. First is specimen. Specimen is what's on the slide. If I'm looking at a slide of lung tissue, then my specimen is lung tissue. The field of view is what you can actually see of the specimen when looking in the eyepiece. Now that's not the entire specimen, just the part you can see when you look into the microscope. Magnification refers to the number of times bigger than real life that your specimen appears. Now how do we calculate magnification? It's always the eyepiece, which is always going to be for us 10x, or 10 times normal light, times whatever objective we're looking at. So for example, if I'm looking at something under the 40x objective, my total magnification would be 10 times 40, or 400x. Notice how I always put that x back there, right? That's very important. And the last one is scale. Scale is the number of times bigger or smaller a drawing can be than real life. So a drawing can be 10 times bigger than real life, or half a time bigger than real life. Notice that we never use fractions in science, only decimals. So if this was real life, right, this would probably be a 5x figure, and this would be a 0.5x figure. Alright, so let's move on. Let's talk about some basic biochemistry now. Again, these are things that you should have gone over last year. If you are new, this is just a quick review, and if you have any questions, of course, you can ask myself or Mr. Kell, or even Miss Baden if you feel more comfortable with that anytime you need. Alright, so element is the smallest unit of matter that still retains an individual characteristic, meaning that it has unique properties like gold, calcium, sodium, etc. These are the things that are found on the periodic table. There are 92 naturally occurring elements, 118 elements that we know of. The rest are made in laboratories. When you have two or more elements that are actually chemically combined, that is called a compound. So, for example, water is not an element. There is no water on the periodic table. But there is hydrogen and oxygen. When I bring them together in a chemical reaction, I get H2O. The important thing to note about a compound is that a compound has different characteristics in the elements it's made of. Perfect example is water. Oxygen is a breathable gas. Hydrogen is a flammable gas, but if you put the two together, you have a liquid, water. When you have two or more elements coming together in a way that chemically changes them, that's called a reaction. Right? It is important also to realize that reactions involve energy. Either the reaction takes in energy or it releases energy. Below there is one of the most important chemical reactions in biology. This is, anybody recognize this one? Hopefully you guys recognize this as the photosynthetic equation. Meaning that water and carbon dioxide will yield glucose and oxygen. Now, we're right smack in the middle of biology. And biology, by definition, is the study of life. So, if we're going to study life, we need to figure out what the requirements for life are. Now, most scientists around the planet recognize 14 different requirements for life. We're not going to talk about all 14. We're just going to talk about a few of them. Some of them get a little nitpicky in terms of evolution and ecology, and we're just not ready for that. What I want you to know about the requirements for life is that 1, all living things must grow and reproduce. That doesn't mean they have to. It means they have to have the ability to grow and reproduce. All living things must respond to stimuli, meaning outside, light goes on and I go, the light is bright. Or you put sugar down on the ground, ants sense it and they'll head towards it. Put acid in a petri dish, bacteria will move away. Those are all examples of outside stimuli. Put a plant in a sunny window and it'll start growing towards the light. Again, that's a response to an outside stimulus. Living things also respond to inside stimuli, inside reactions. We call that homeostasis. Homeostasis is Latin for body stop. So what that means is that the internal environment the internal environment is constant. Things like your blood pressure, your body temperature, your blood volume. All those things are things that your body regulates. Your blood's pH. All those things. All living things must use energy, and the energy that living things use, the battery that powers us, is called adenosine triphosphate, or ATP. It is the whole point of the photosynthetic cellular respiration pathway. We'll talk about that in just a second. All living things must be made of cells. Cell must be the basic unit, and it must contain DNA as its instructions for it. Again, when we're talking about living things, I just said that living things must be organized into cells. So what I have here is a hierarchy of what we call organization. We're going to talk a lot about this year, so everybody's listening up. An atom is the smallest unit of an element. It by itself is not alive, because an atom by itself can't do the things that we just talked about on the previous slide. A molecule is not alive for the same reason. So if I get a whole bunch of atoms working together in a compound, that one unit of that compound is called a molecule. If I make a really big molecule by putting together two little molecules, that's called a macromolecule, which is macro is literally for big. If I put a whole bunch of macromolecules together, I'll have what's called an organelle. Remember, an organelle is a part of a cell. A whole bunch of organelles working together is called a cell. At this point, we officially become living. From there, cells can actually work together towards a common goal. We call those tissues. Tissues can work together to form organs. Organs can work together to form organ systems. And organ systems come together to form organisms. So let's do this. I'm going to do this with two different examples. Let's first take the example, just a one right out of science. You can have one atom of carbon. You can have a molecule of glucose. You can have a macromolecule of starch, which is many glucoses together, kind of like that. You can have a whole bunch of macromolecules of starch come together to form the organelle, the vacuole, where starch is stored. Okay. Then you can have a cell. It has a whole bunch of different organelles in it. Now look what we've done here. Every one of these is still carbon. It's just carbon as part of this, as part of this, as part of this, as part of that. So then you have a whole bunch of cells together that form a tissue. Then you have a whole bunch of tissue layers that come together to form an organ, like organ like the stomach or the heart. Then you can have, so this is my heart now. And then you can have an organ system like the circulatory system, which is the heart, all the veins, all the arteries, the lymphatic system, the lungs, like that. And then together they all form an organism. That's you. So if I break it down, you can actually see that it still just started with one atom, and it built up all the way to something bigger. So what we're seeing is just little things coming together to organize themselves to do something greater. Let's talk just for a second about cells. As we just said, cells are the smallest unit of life. All cells, by definition, no matter where they are, no matter what organism they are, bacteria, humans, foot cell, heart cell, brain cell, whatever, all cells have a phospholipid membrane, a phospholipid bilayer. This is so important. There are some people who argue with me, they're wrong, but there's some people that will argue with me when I say that the membrane, the phospholipid membrane, the cellular membrane, is the most important part of the cell. Again, people argue with me, they are wrong, I am right. Because it is through the phospholipid membrane that cells actually talk to each other. And decoding the language of cells is the key to all disease. I mean, what is diabetes but the cells of the pancreas not talking to the rest of the body anymore? What is paralysis? When people are paralyzed, it's because their spinal cord cells don't talk to the body anymore. If we could somehow decode that language and get the parts of the body that are sick to start talking to each other again, imagine what we can do. Other parts of the cell that I want you guys to know is the ribosomes. Ribosomes make proteins. That's going to be very important later on. The DNA is found in the nucleus. That is the genetic material. It is essentially the blueprint or the recipe book for the entire body. And the mitochondria, because the mitochondria make energy, that ATP molecule. And it is here with what the mitochondria does that I'm going to spend a little bit more time in just a moment. Let's take a look at the types of cells. There are two basic types of cells. First are called prokaryotes. Prokaryote is Greek. It means before chromosome because it doesn't have a defined nucleus, right? A prokaryote is important because there is no nucleus. You do not need a nucleus to have a cell. You do, however, need a cellular membrane. So this is your basic prokaryote. These are things like bacteria and archaeobacteria. As you can see here, there is no nucleus. There is DNA, but it's all kind of coiled together in the middle in a circle. If I were to unwind that, it would look just like a circle like that. Eukaryotic cells, on the other hand, are much more sophisticated. There is a nucleus. There are sophisticated organelles such as lysosomes, endoplasmic reticulum, Golgi apparatus, nuclei, and vacuole. I will let you guys read through this. Again, you should still have your notes from last year. Please review the organelles and what they do. This is what your typical animal cell looks like. As you can see, it's much more complicated than the inside. You have a nucleus in the center. If I were to see the DNA, it would not look like a circle anymore. It would look like an X. And we'll talk a lot more about what that means in second module. If I were to look at the difference between an animal cell and a plant cell, the first thing that would come to my immediate attention is that an animal cell is round and a plant cell has more of a square box-like structure. In fact, most animal cells are not round, but it proves the point because most plant cells are boxy shaped, and that is because of this organelle right here, the cell wall. Plants have a cell wall, animals don't. And they need it because plants don't have bones. And yet, trees are sometimes 5, 6, 7, in some cases 20 or 30 times taller than a human child who has bones. And so you got to figure, how is that plant going to stand straight up into the sunlight, because that's where its food is, if it doesn't have bones? Well, it's got to do something. So it uses these cell walls to maintain its structural stability. Other differences between a plant and an animal cell is that a plant cell, you can see, has a very large centralized vacuole, whereas an animal cell does not. And plant cells have these little guys here, chloroplasts. Chloroplasts look a lot like mitochondria with a few differences, and their job is to make glucose. Because they can make glucose, they are known as autotrophs, as opposed to us, which are heterotrophs. Going back to the animal cell real quick, I can see them here, right here, these centrioles, those are something that only animal cells have. So animal cells have certain organelles that plant cells don't have, and plant cells have certain organelles that animal cells don't have. All right, the last thing in our review that we're going to talk about is ATP and energy. And so what you're looking at is a very complex picture of ATP. Adenosine, which is what this is right here, triphosphate, 1, 2, 3, 3 phosphates. It is actually right here in the chemical bond between the this is alpha, beta, this is gamma, between the gamma phosphate and the beta phosphate. This is where all the energy is held. Very, very, very important bond. As this bond breaks, energy is released. As this bond comes back, energy is put back together again. Now, do I care if you know all that? No. All I want you to know is that biological energy is called ATP, and that ATP has 3 phosphates. So how is ATP made? This was, I believe, the last thing we did last year. There is a 2-step process to making ATP. Right? Step 1 is photosynthesis. Right? This is where we actually take the energy of the sun and turn it into something that a human being can use, or any organism for that matter. The way I describe it is, let's say you won the lottery, and you won $1 million, trillion, billion, and you were wealthy beyond your wildest dreams. But the catch is, they paid you in gold bars. So imagine it. Imagine the biggest room in your house filled to the absolute brim with gold bars. Awesome. Yeah, I know. Okay, but here's the problem. Let's say you want to go to the movies with your friends, and you're going to take the entire 8th grade class to the movies. You're rich, you can afford it, so you go for it. But you have a gold bar. So good luck bringing that gold bar down to the movie theater and saying, hey, can I have, 101 tickets to the show? They're not going to take that gold bar. That's the problem with with the energy from the sun. The sun's energy that hits us is enough to power our entire world for free forever. The problem is we don't have a way to convert that energy into something that biological organisms can use. The only person that can actually change those gold bars into dollars or change the sun's energy into biological energy are green plants. So step one is photosynthesis. That's when we take solar energy, put it into glucose. This happens in chloroplasts and in chloroplasts only, and the protein chlorophyll is incredibly important. A lot of people get confused between chloroplasts and chlorophyll. What I always used to say is that chloroplasts are filled with chlorophyll. That kind of Then the glucose, which is a type of sugar because it ends in OSE, is then taken in by animals, brought to the mitochondria for step two. Step two is called cellular respiration. All living things do cellular respiration, even plants. Right? There's this myth that plants don't have to do cellular respiration because they do photosynthesis. It's not an either or. It's a two-step process. Plants are just able to do both steps. Humans and other animals have to rely on plants to do step one, and then we show up in step two. That's when you take glucose to ATP. It occurs inside the mitochondria mostly. It requires oxygen for its final step. If there is no oxygen present, you're working out or something like that, you just don't have enough oxygen, there will be no cellular respiration. Instead, you have to go to plan B, which is called fermentation. And that is when ATP is produced without oxygen, producing lactic acid in mammals, and alcohol in other organisms like yeast or bacteria. Okay, so that was our review. I hope you enjoyed it. Watch it as many times as you feel like you need to, and answer the questions in your homework packet under homework 1-1. Thank you very much.